gain weight, vomiting. There was no lethargy or convulsion coma in this child. So we have two pathways to go. One is the metabolic disorder. The other is our common infection, traumas, any CNS anomalies and all. So metabolic disorder, we obtained a plasma ammonia for this child, which was high in this child. So from the flowchart, then you have to obtain the blood pH, CO2 and HCO, your bicarbonate. With the normal anion gap, it can be urea cycle defect. If the high anion gap, it can be acidosis, that is your organic acidemias. And so normal anion gap with the normal ammonia or aminoacidopathies or galactosemia is the differential. So our evaluation, we got our TMS and GCMS back. In that, we have a very high methyl malonic acid in blood and urine. So you can see here in this slide, your methyl malonic acid levels are very high. So we proceeded with the gene study to confirm the diagnosis. In this gene study, MMA plus gene, that is this gene coding for MMA protein, was abnormal in this child. So the disease of this pathology is methylmalonic aciduria of the CPL-A complementation type. That will be discussed in detail by our biochemistry department, and that is pathogenic in this child. Okay, so we proceed with the treatment. Treatment is multivitamin supplementation, especially your cobalamin, it is vitamin B12, and your levocarnitin, and 50 to 100 mg per kg per day as a dose we are supposed to give. And we gave a restricted planned diet for this child, and we offered the parents genetic counseling in case they are planning for another pregnancy. They should be planned because another child should not have this disease. And this is the diet chart we gave to the baby. And this is with the help of our dietary department. This will be discussed in detail from the dietary department. And for the course, the child is now thriving well with adequate weight gain. His tone improved. There is no hypertonia now, and new milestones are achieved now. From the review, we found out that the child is now able to sit without support. So no episodes of vomiting. His present weight is 7.35. So this is his growth chart now. So you can see that from he was falling in between, now he picked up and he is growing well. So I don't have anything. So this will be discussed in detail by biochemist. Thank you.
Thank you very much, Dr. Sujin Mariam Thomas. That was indeed an excellent presentation. We will take questions to Dr. Sujin Mariam at the end of the session. So welcoming one and all once again to the second part of the presentation, Biochemistry of the Disease, a case of methylmalonyluria. This topic of second MBBS will be taught to the students by Dr. Riju Matthews. He is currently, he did his MSc Biochemistry from CMC Vellore and PhD in Biochemistry from Yanapoya University of Mangalore, and he has 23 years of teaching and lab experience behind him. He also has a certificate holder in law in hospital administration by CMAI, and he is currently working in the capacity as assistant professor in Department of Chemistry in Believers. And he is behind the NABH and NABL accreditation for our lab. And because we have all noted that because of him only, we have zero error from the Department of Biochemistry so far. So we physicians, pediatricians, surgeons, we all have to agree that we are really appreciating him for his services to Believers. Over to you, sir, for your presentation.
Good afternoon, everybody, sir and madam. Today I will be presenting the biochemistry of methylmalonic aciduria. It is one of the commonly seen organic aciduria in our among the children population or the newborns. Let us see the chemistry first. How do we get organic acid in our body? It's basically from amino acids, neurotransmitters, carbohydrates, microorganisms, fatty acids, purines and pyrimidines, cholesterol, as well as from drugs and special diets. Also, let us see what is this basic of this particular term called methyl malonic acid urea. The term urea
Psalm 27 towards 7 onwards. യഹോവേ, ഞാൻ ഉറക്കെ വിളിക്കുമ്പോൾ കേൾക്കണമേ, എന്നോട് കൂടെ ചെയ്ത് കുറ്റമല്ലെങ്കിൽ എന്നെ മുഖം രക്ഷിക്കുകയും എന്റെ ജീവിതത്തിൽ കൽപ്പനമൊന്നു എന്ന് എന്റെ ഹൃദയം പറയുന്നു. യഹോവേ, ഞാൻ നിന്റെ മുഖം അന്വേഷിക്കുന്നു. നിന്റെ മുഖം എനിക്ക് മറക്കരുതേ, അടിയനെ പോലെ എനിക്ക് കളയരുതേ. നീ എനിക്ക് തുണയായിരിക്കും.
And you know, when you see Mia that is in blood, when Aspermia is in,
occasionally hospital
the organic acid urea first chain will be in MIA. Here, then it will be present in urine, so it is known as urea, URIA, not UREA. So methyl malonic acid in urea. Whenever we say acid in organic chemistry, it's COH, 3 double bond O, single bond OH. And now also see what is that chemistry regarding this methyl malonic. Now looking at methyl malonic, first I remember when I studied +2, there was dibasic acid that was discussed. These are the list of dibasic acid: oxalic acid with 2 carbon, malonic acid with 3 carbon. So I am stopping here. Malonic acid has a structure of COH, CH2, COH. Then it goes like that, which has the mnemonic of oh my son, go and pray silently and solemnly. So that are all dibasic acid that comes. Then looking at malonic acid, you see it is COH, CH2, COH. When a hydrogen from the CH2 is removed and a methyl group is added, that will become methyl malonic acid. So the hero of the story is methyl malonic acid here. We'll go have a small review of amino acids. This, as we know, there are 20 standard amino acids. Some of them are essential and some of them are non-essential. So when you classify them based on the side chain, they are either aliphatic, aromatic, acidic, basic, hydroxylic, or sulfur-containing or amidic in nature. So we will see some of these amino acid names again in the next slide. We'll come back and see what they are. So this is the propionic acid and methyl malonic acid urea pathway, which is sometimes or which is always called vomit pathway. We also see in our case study that the baby had a vomiting, recurrent vomiting. So this is called vomit pathway. Vomit pathway stands for valine, odd chain fatty acid, methionine, isoleucine, and threonine. So how these are connected? See, these all metabolism is connected together to reach to methyl malonic acid and to succinyl-CoA, then to TCA cycle. We'll see how it is happening. Valine and isoleucine, then threonine, methionine, odd chain fatty acid. So all together they will come as vomit pathway. Remember this: valine, odd chain fatty acid, methionine, isoleucine, threonine. If you remember this valine, isoleucine, they are the aliphatic amino acids. Valine here and isoleucine here. What are they? They are essential. That means this protein, this amino acid has to be in the diet. Then comes the methionine, which is the sulfur-containing amino acid, again essential in nature. Then comes the threonine. Threonine is again the hydroxy amino acid along with serine, but the threonine is the essential amino acid. So this pathway consists of essential amino acid, but the odd chain fatty acid is very rare in our diet, in our metabolic pathway. But usually this is connected. So propionyl-CoA can be created. A biotin dependent propionyl-CoA carboxylase is required for it to convert it to be methyl malonyl-CoA. From methyl malonyl-CoA, vitamin B12 dependent methyl malonyl-CoA mutase is required to be converted to be succinyl-CoA. When this methyl malonyl-CoA mutase is absent, that will lead to built up of methyl malonyl-CoA in blood, that is called methyl malonyl acidemia, then as urea. So we will little bit look into the other details also, including these details we already told. The propionyl-CoA to conversion to methyl-malonyl-CoA requires biotin B7, and the methyl-malonyl-CoA to succinyl-CoA requires B12. So the result will be the build-up of all the precursors, methyl malonate, sometimes methyl citrate also, that being acid, that will end up in acidosis also. When you look at methyl malonyl-CoA mutase, 60% of cases are the result of the mutation in MUT gene, which encodes methyl malonyl-CoA. But there can be also the action of this enzyme can also be crippled by mutation in methyl malonyl acid A, MMA B and MMA DHC genes, each of which are encoding protein that are required for normal functioning of methyl malonyl-CoA mutase. So what are they? Again, going to details of that. The first one is MMA A, that is called cobalamin A in chromosome 4, that codes for mitochondrial methyl malonic acid urea type A protein, which is involved in the translocation of cobalamin into the mitochondria. If you remember the slide which was previously shown, this is the gene that is actually having some abnormality in this particular case. So the second one can be cobalamin B in chromosome 12 encodes for the enzyme that catalyzes final step in conversion of vitamin B12 to adenosylcobalamin. This adenosylcobalamin is the cofactor that is required for the catalytic activity of the enzyme mutase which we were talking about. The third one can be MMA ADH, which is in chromosome 2 and encodes for mitochondrial methyl malonic acid urea and homocystinuria type D protein. And this is also seen when you have a methyl malonic acid and homocystinuria co-present in that babies, which is not in the case in this particular cell. So this is the result. You can see MMA-A is the genetic defect that is pathogenic in case, which is autosomal recessive in nature. And this is the area where the vitamin B12 that need to be transported into the mitochondria in the adenosylcobalamin. That is a coenzyme for the methyl malonyl-CoA mutase, which converts methyl malonyl-CoA to succinate. Succinate is absent in this particular case or abnormal in this particular case. So this is a complete metabolism of cobalamin that is involved in homocysteine to methionine conversion as well as the methyl malonyl-CoA to succinyl conversion. You can see multiple conversion of cobalamin vitamin B12. So looking at the clinical presentation, this has been already discussed. I am not telling, I am only mentioning that vomit. All other things which has been already discussed. I only want to tell you about the ketoacidosis presentation. That's a blood ketone. We have in laboratory blood ketone detection, that is beta ketone, beta hydroxybutyrate from capillary blood, which is a point of care testing with a biological reference range of 0.0 to 0.6 millimoles per liter. And urine ketone, that's part of complete urine analysis, can be done by a urine FIPOR or a Rothras test, usually a random urine sample, and the reference range is negative. Looking at ammonia, which is a key finding in this particular case, is measured by the spectrophotometric assay in AEC 480-680. Specimen is a fresh EDTA plasma. This you have to remember that after collection of immediately, this sample has to be placed on ice, centrifuge in cold, and sample has to be as soon as separated from the plasma and has been stored at 2 to 4 degrees or frozen immediately. Ammonia samples are stable for 3 hours at 2 to 4 degrees and 24 hours at -20 degrees. So the biological reference range is 10 to 47 micromoles per liter. And the last test that is done in our laboratory, next one is vitamin B12, which is commonly available by chemiluminescence assay, which can be measured in picograms levels, done by Magliumi 1000 and 2000. Specimen is serum, and the biological reference range is from 2000 to 200 to 1100 picogram per ml. And that's all. Thank you.
Yes, thank you, Dr. Riju Matthews, for that wonderful presentation. So we are expecting questions to Dr. Riju Matthews at the end during the moderation. Thank you so much, sir. Due to lack of time, we are proceeding to the next class, that is dietary management in organic acidureas. This class will be presented by our chief dietitian, Ms. Jyoti S. Krishnan. She is chief dietitian and she is a PhD research scholar with 15 years of experience. Before joining Believers, she worked as senior dietitian at Amrita Hospital, Kochi. She is a life member of Indian Dietetics Association and Nutrition Society of India. She has published many articles on diet and nutrition in various national and international journals. Once again, we request the students and attending faculty to make this an interactive session. Please post your questions in the chat box. All doubts will be cleared at the end of the session during moderation. Over to you, Ms. Jyoti S. Krishnan.
Good afternoon, one and all. Today, I'm going to discuss about the nutrition management of methylmalonic acidemia. As all of you are aware that in methylmalonic acidemia, the body is unable to break down the amino acid methionine, threonine, isoleucine, and valine. And as a result, the methylmalonic acid is built up in the blood and tissues. So the primary management of methylmalonic acidemia includes a low protein diet. That is, the diet should be continued for the lifelong. So the diet should provide adequate amount of proteins. The same time, it should not build up these particular amino acids in the blood. So the diet nutrient requirement was calculated as the anthropometry. Already it was discussed by Dr. Sujin, and the weight of the child was 6.5 kg. So I have calculated the calorie of the child as 120 kilocalorie per kg of current body weight, and the protein was usually the 8-month-old child, the protein requirement is, as per ICMR 2010, it is 1.69 gram per kg body weight. But this case, we have restricted the amount of protein and restricted all the amino acids like methionine, threonine, valine, and isoleucine. So the easy method to calculate the nutrient requirement was using a protein exchange. Protein exchange means we have to take the protein content of each food and we have to use the exchanges for selection of the food. So it means that if the protein content per portion is 0 to 0.3 gram, that is very low amount protein, it should be considered as a free food. And if the protein content of the food is 0.4 gram to 0.7 gram, that means an average of 0.5 gram. That will be considered as a half exchange. Then if the content is 0.8 gram to 1.2 gram, then the exchange will be 1. And if the protein is 1.3 to 1.7 with an average of 1.5 gram, then that protein exchange will be 1.5. So it is a very easy method, because otherwise it will be very difficult to calculate all the protein content per food, and to introduce the diet will be very difficult. So after calculating the protein exchange, actually it is a very easy method of calculating protein requirement, because it is very easy to categorize the foods based on the colors. Because in the first category, I have included these food items as green foods, which are considered as free foods, free protein foods. So in these foods, there are no low levels of methionine, threonine, valine, and isoleucine is present. So mainly I can say that the category of fruits, almost all fruits are low in all these essential amino acids, and almost all tubers means those food items which grow under the soil. That means tapioca, then beetroot, carrot, all these comes under the tubers, and which are considered as free food. So then in the vegetable group, if we take cucumber, radish, or pumpkin, that variety of food items like cucumber and all, it is included as free food. And in the sugar category, jaggery and sugar is also containing very low amount of methionine, threonine, valine, and isoleucine. So again, in the green food group, there are some foods which can be prescribed in particular amounts. If we take it more quantity, it can be considered as an exchange. So for example, if 1 banana per person is allowed as free, but if we take 2 banana, it should be encountered as 1 exchange. So that gives 1 gram of protein. If you take 2 banana, it will give 2 gram. 1 gram of protein. That is one exchange. Then some other foods like in the cereal group like sago, then arrowroot, etc. are considered as the free food. If taken in more quantity, that will be encountered as one exchange. Then the same with the case of mushroom. If we take 5 mushrooms a day, that can be taken as free. And if it is 10 mushrooms in a day, it should be counted as one exchange, that is equal to 1 gram of protein. So all the food items, it is possible to convert it into exchanges and can be incorporated in the menu. So the second category, as I said, I have included 3 category of foods. In the green category, it is very easy to incorporate that food items in the diet because it is very low in protein. So in the orange category, the food items can be provided to the patient, but in a measured amount. Like if you take butter, 1 teaspoon of butter itself give 1 gram of protein. So if you take it more quantity, that will increase the protein content of the food. So similarly, normally prescribed items like rice, wheat, oats, suji, etc. If we take half cup per day, it is encountered as 1 gram of protein. So similarly, in case of pulses, it is very high in protein, as all of you know. So even if very small quantity, it can give a more protein. So a minimum amount of 1 tablespoon of cooked weight of pulses and dals will provide 1 gram of protein. So if we take any food item from this orange category, 1 portion which will be equal to 1 gram of protein. If it is taken 2 exchanges, it should be encountered as 2 gram of protein. If it is 3 exchange, it should be 3 gram of protein. Similarly. So the third category is the red category. That is, it is better not to use these food items. And if it is taken very small amounts also, that will cause more amount of protein in the diet. So in case of non-vegetarian food items like chicken, fish, egg, etc., even if we take a 10 gram portion, it will give 1.5 grams of protein. So in case of milk, if we take 50 ml, that itself give 1.5 gram of protein, and cheese, even 5 grams will give 1.5 gram protein. So it is better to avoid all these food items. It is better to avoid, but if we use it, it should use cautiously. So the next thing is understanding the synthetic protein. If it is not possible to meet the calories and proteins with the prescribed menu, it is possible to achieve the calorie and protein requirement with the help of synthetic protein. Synthetic protein is a specially made protein that contains all the amino acids except methionine, threonine, valine, and isoleucine that the child needs to grow. And the synthetic protein also contains some essential vitamins and minerals. So that if we recommend the synthetic protein for the child, it is better to incorporate in similar amounts in the diet. And if we incorporate it in the diet, it must be taken for the lifelong. So after a proper planning of the diet only, we have to incorporate the synthetic protein. In this child's case, actually, I was able to meet the protein and calorie requirement without a synthetic protein supplement. So in case of an adolescent or a toddler or some school-going children, it may be very difficult to meet the protein requirement with this particular restrictions. At the same time, we can incorporate the synthetic proteins in the diet. So actually, why I have selected all these food items is because most of the time the recommendation of methionine and isoleucine in the diet, actually we have to, first of all, we have to check the recommendation of isoleucine, all these amino acids. So it is in case of methionine and isoleucine, the recommendation is 30 milligram per kg body weight, and in case of threonine and valine, the dietary recommendation is 34 milligram per kg and 39 milligram per kg. So if we think about a non-vegetarian diet like egg or chicken or fish, that itself will give, even a 10 gram portion that I have included earlier, itself will give almost 100 milligram of these amino acids. So when we plan the diet, it will be very difficult with all these non-vegetarian, like a high protein content foods, to meet the nutritional requirement of the child. So in this menu, I have included the items which are from green and orange category, and I was able to meet the calorie and protein requirement of the child. And initially, we have started with 120 kilocalorie per kg calories and protein 1.5 gram per kg, which gives around 9.75 gram protein, and carbohydrate was given as 65% of the total calories, that is equal to 130 gram, and fat was 30% of the total calorie, and that is equal to 26 gram. So in the menu, in the early morning and breakfast itself, I have included 2 exchange of proteins, and in the lunch and mid morning, I have included 3 exchange of proteins, and tea time like 4 p.m. 1 exchange of protein, and 6 p.m. and dinner I have included 3 exchange of protein. That is altogether 9 exchange of protein is used in this menu, and the child was able to meet 9.75 gram of protein. So I hope all of you clear with this slide, and if any doubt or any suggestion regarding this, please let me know. Thank you.
Okay, ma'am, thank you so much. This is 800 kilocalorie, low fat, low protein diet, which is restricted methionine diet. Okay, we have to come back to Dr. Sujin Mariam Thomas. Dr. Sujin, can you please continue with your case? Actually, what happened to this patient which was in question? Please come back to the patient. This patient was in the ward, and actually what happened to the patient? How did you manage this patient with methylmalonic acidemia?
Okay, thank you, madam. This patient, we gave the diet just now explained. We gave a low protein, low fat diet with all the protein adjustment, and the child did absolutely well and is now slowly gaining the milestones and catching up with the growth. So, and vomiting episodes are decreased, and apparently he's accepting foods, and there is no vomiting, and the compliance is also good. And the child is now happy with the new milestones. He's, as I told earlier, he's able to sit without support now. Now he's around 8.5 months old. And so at this time, I want to tell you why this is important, because you have heard a lot from the biochemist. Riju sir has told you anything about methylmalonic acidemia, and our dietitian explained clearly about how you should plan a diet. But why this is very important? Why you should know all these things about methylmalonic acidemia? Yeah. Okay. Theoretically, methylmalonic acidemia is rare. That is 1 in 50,000 to 1 in 1 lakh live births. It is the incidence reported. But you know, this is my second case of methylmalonic acidemia. Once when I was doing my PG, and the second is this. Once was already a diagnosed case, and this is not diagnosed. That we were fortunate to diagnose. So why, as a medical student, why you should know about this methylmalonic acidemia is because if we don't treat the child, that child will end up on garden variety of mental retardation, and there is no going back. So once you miss the case, there is that is like you are spoiled, like that life is not fulfilled as right now. If the child is going like apparently going like this, the child will be able to do like whatever he is capable of. But if we would have missed the case, that child would have landed up as a case of mental retardation. So this is very important. This is one of the preventable cause of mental retardation in our pediatric cases. So it's so now you may understand that why you should diagnose this case and why it is very important. So methylmalonic acidemia is basically the elevation of methylmalonic acid in blood and urine. I'm not going into detail because you already know everything. The average age of clinical presentation is 6 months. So the problem with the diagnosis is it can have a varied presentation. Like in our case, the child just presented with vomiting. Like the mother's primary complaint is vomiting, and the developmental delay and hypotonia and all, we from our history and examination, we found out that that was not the complaint of the mother. The mother came to us with only vomiting, and the child was actually referred case, and referred to us for evaluation of vomiting and failure to thrive. So vomiting is very common in pediatrics, and failure to thrive is also very common. So the first thing you evaluate in a child with vomiting is not something like inborn error of metabolism. But you should keep that in mind, because if you diagnose the case at correct time, you can treat the child and the child can do well. So methylmalonic acidemia, from the mechanisms that Sir explained, it can have a wide variety of presentation. Like the child can be sick at birth, or the child can be like apparently well till adulthood. So uh, uh, with a high level of suspicion only, you can do the test and diagnose the case. So the average age of clinical presentation is 6 months, we take an average. Mean age of diagnosis is 24 months. It can occur alone or in combination with other biochemical abnormalities. And there can be other abnormalities like homocysteine, low methionine. It's already described. And it can be due to either the methylmalonyl-CoA enzyme or a vitamin B12 transport, or apparently vitamin B12 deficiency can also cause methylmalonic acidemia. Okay. And severe forms can have lethargy, vomiting, seizures, feeling problems, tachypnea, hypotonia even in the first few days of life. But less severe forms can be asymptomatic up to adulthood. It usually present during the attack. The attack is like when the infant is like in prolonged fasting, or there is an infection, or a high protein diet. All these can stimulate an episode of this catabolic state and a high, or we can trigger an attack. So this attack can lead to many grave complications like stroke, like in the basal ganglia area, they can have an attack and it can lead to permanent damage. And multiple attacks like this can leads to like your complications like tubular anesthesia, that is a renal failure, hepatic failure, pancreatitis, and all these like this multiple attacks coming into this child can go for a hepatorenal failure also, that meaning a renal and liver transplantation. So it is better to avoid these attacks than go and treat the attack. So you should counsel the parents very well that in any case of infection or fasting or high protein diet, all these can trigger an attack. Even in apparently normal, who is diagnosed with methylmalonic acidemia and is otherwise doing well, but in these situations they can have an attack. So these attacks can lead to all the complications. So multiple attacks slowly add up and lead to chronic, like the problems also will be chronic, and you will land up in irreversible damages. So, and this mild variation, this now a child, it is a mute, that is mute negative. That is there is some not like completely absent. There is some enzymes are there, and it's a cobalamin A type variation. So that is better than the cobalamin B, C, and the other types. So this child is fortunate to have this kind this problem, because that is compared to the other things, it is it has a better prognosis, and it's like we can treat it, because some enzymes are there. If we provide the diet and if we prevent all these triggering factors, this child can do well. Okay, so mainly the treatment is diet, supportive medication, and the prevention of all the catabolic state. And the complication already told you: recurrent pancreatitis, growth delay, intellectual disability, optic nerve atrophy, and if the child neglects all the dietary advice and all, and keeping on having a high protein diet, or can have a hyperammonemia state, it can lead to encephalopathy and all the complication of hyperammonemia. And I told you about the stroke. It is a metabolic stroke during an acute episode of metabolic decompensation with a basal ganglia injury. That can also lead to irreversible damage. And take home messages: you should have a high clinical suspicion in diagnosing IM, because it can present like any other gastroenteritis infection like that. So if you keep this definition in your mind when you evaluate a child with vomiting and failure to thrive. And 1/3 of pediatric mental retardation cases may be attributed to the inability to detect a metabolic disorder in early childhood. So rate of occurrence is high in our country because the population is more. And this case emphasizes the importance of clinical awareness and accurate biochemical analysis in diagnosing IM. Thank you.
Okay, that was indeed fantastic, Dr. Sujin Mariam. And this is really fantastic, because we have learned so many things about inborn errors of metabolism, that is IEM, methylmalonic acidemia, phenylketonuria. Then we have homocysteinemia. There are so many inborn errors of metabolism. And now we have a last session, in which we have a surprise faculty, Dr. Cletus CC, Assistant Professor in Biochemistry, with a few rapid fire questions for you on organic acidureas. So be ready. with your answers in the chat box. Are you ready, children? Say yes. Yes? Okay. So you have your favorite Cletus sir asking you questions.
So good afternoon, everyone. So now I'm going to ask some questions generally from the organic acidureas, not specifically for the methylmalonic aciduria and protonic aciduria. My first question is related to methylmalonic aciduria. Name the CBL variant which cause methylmalonic aciduria alone. So what is the answer for that? So you can see the figure here. In order to answer the question, you should know this pathway, how cobalamin is converted to the adenosylcobalamin, active form of the coenzyme, which is required for the methylmalonyl-CoA mutase. Once the cobalamin, which is received by the receptor with transcobalamin, it is carried by transcobalamin. Once the receptor, CD325 receptor, will take into inside the cell's cytoplasm, and it will move to the lysosome. There, it will be released from the transcobalamin. So then the free cobalamin will be coming out from the lysosome, and it will combine with another protein called MCACHC. So this protein will reduce the valency of the cobaltin from 3 to 2. You can see 3 to 2. And this cobalt 2 will be further available to addition for the methyl or adenosyl group. So the methyl group will be going for the methyl cobalamin. It will combine with the methyl cobalamin. So this methyl cobalamin is a coenzyme which is required for the conversion of homocysteine to methionine by the enzyme methionine synthase. So other portion of the cobalamin will be moved into the mitochondria, and further it will combine with the adenosine to form adenosine cobalamin. So adenosine cobalamin is the coenzyme for the reaction methylmalonyl-CoA into succinyl-CoA by the enzyme methylmalonyl-CoA mutase. So here the question is the name the CBL variant which cause methylmalonic aciduria alone. You can see CBLA and CBLB and the CBL2 V2 variant. That is the first option. And the second one is CBLE, G, D and V1 variant. And CBLF and CBLJ and CBLC, that is the third option. So here the answer is A. Here, this is actually very important to pinpoint the disease. Actually, now the case which is presented by the doctor has pointed out the diagnosis. The case is a CBLA way complement type. So here, this is important. Actually, if there are any CBLC or CBLD variant, V1 variant, it would have been caused both methylmalonyl-CoA aciduria as well as homocystinuria also will be manifest. Then the second question is, this question is used to ask in competitive exams. So this is very important, I think. So sweaty feet odor, which is seen in what condition? So the first option is maple syrup urine disease. maple syrup urine disease, you all know that it is due to
No explanation.
okay.
Phenylketonuria,
Yes,
multiple carboxylase.
multiple carboxylase, then isovaleric acidemia. So
Cinderella has answered as a D.
yes, that is correct. So here, that isovaleric acidemia is characterized by the deficiency of the enzyme isovaleric CoA dehydrogenase enzyme. So isovaleric CoA and its metabolites will be it appear in the sweat and in the cerumen. So that will the characteristic order is sweaty feet order. The name of the vitamin is required for the propionyl CoA carboxylase. The name of the vitamin which is required for the propionyl CoA carboxylase. Answer is B.
B is what everyone is asking.
Yes, B is biotin. Most of the carboxylation reaction, biotin is the coenzyme. You can see the pyruvate carboxylase, propionyl CoA carboxylase, then acetyl CoA carboxylase. Like that, all enzymes which is required, biotin as the coenzyme. Then what are the clinical characteristics of methylmalonic aciduria and propionic aciduria? Except the option A is hyperammonemia. Hyperammonemia. Second one is metabolic acidosis.
And see, Pasna, Adira, Tara, Drishya, all have answered C.
Yes, yes, your answer is correct. So definitely there will be a high anion gap because there is an acidosis. So the bicarbonate concentration will be depleted. That will lead to wider anion gap. Then question number 5. Hyperammonemia inhibits the TCA cycle by depleting what? First is oxaloacetate. Second is alpha Kg. Third one is citrate. Fourth one is succinyl CoA and fumarate. What will be the answer for this?
They answered C as the answer. No, D also something.
No.
D, B. Many people answered B. Angel,
Anyone asked answered alpha ketoglutarate?
D and B.
B there and answer.
C also something.
Yes, the answer is B, alpha ketoglutarate.
B
Why?
is very only one or two. Anaga and
Yeah, I will tell you the explanation for that. So in hyperammonemia, as part of the homeostasis, body will try to reduce the ammonia level. Actually, this glutamate will be combined with one ammonia molecule to form glutamine. So the source of the glutamate is alpha ketoglutarate. So the alpha ketoglutarate will be converted to glutamate. So definitely there is a depletion of alpha Kg in the TCA cycle. So there is no replenishment. So here alpha Kg is the answer. Hyperammonemia will inhibit TCA cycle by reducing the amount of alpha Kg. Then question number 6. A baby presents with unusual refusal to feed, skin lesions, seizures, ketosis, organic acids in urine with normal ammonia. What will be the likely diagnosis? The first option is methylmalonic aciduria. Second option is propionic aciduria. Third option is MSUD. D is isovaleric acidemia. And the fifth option is multiple carboxylase deficiency. Multiple carboxylase deficiency means deficiency of the enzymes like pyruvate carboxylase, acetyl carboxylase, then 3-methylcrotonyl CoA carboxylase. So which one will be the answer for this question? You can see this is a normal ammonia. So probable diagnosis actually that
Wait, wait, who is answered? Nobody is answered.
So here MMA, PEA, and multiple carboxylase deficiency, there will be hyperammonemia. So in the case here, you can see the skin lesions. Skin lesions, it is usually associated with the multiple carboxylase deficiency. You can see the skin manifestations, which is appeared in the multiple carboxylase deficiency, mainly due to the enzyme defect acetyl-CoA carboxylase. That skin manifestations mainly attributed due to the multiple carboxylase deficiency. And multiple carboxylase deficiency is actually also related to biotin. During the all carboxylases are biotin as the coenzyme, which is required for their function. Actually, there are two enzymes which is defect in the case of multiple carboxylase deficiency. One is biotinidase and second one is holocarboxylase. These are the two enzymes which is required for the biotin incorporation into these enzymes. Biotinidase actually, which will helps to remove biotin from the dietary proteins, and the holocarboxylase enzyme will helps to form a peptide bond between these enzymes and biotin. So that won't happen. That will lead to multiple carboxylase deficiency. So the peculiar character as per this organogram is the skin manifestations. So this one is like, they will usually as part of the inborn errors in metabolism, usually doctors will prescribe for the GCMS or LCMS. This is a chromatogram for the GCMS. You can see here A is a one condition, B is another condition. Would you be able to differentiate these two conditions? I will give you one clue. Actually, this is one case of methylmalonic aciduria and another one is propionic aciduria. So how can you differentiate which one will be the PA and which one will be the MMA? Okay. See, okay.
B is MMA.
Yeah. Yes. Here you can see the first one. Actually, this GCMS is basically work on, the chromatography, the principle which is working on the chromatography. So here the sample which you are injecting into the GCMS, it will be passed through a column. So based on the affinity, these substances will be separated based on their relative affinity to the column as well as the mobile phase. So here you can see in the x-axis there is a time. So time you can see they have totally run around 20 minutes. So with these minutes, these substituents will be separated based on their affinity to the column. And here you can see in the y-axis the mass by energy ratio. Here, after separating, the mass spec attached to the gas chromatograph will be analyze the sample based on the mass by energy ratio, mass by charge ratio. And you can see there are various peaks. You can see these peaks actually identified by running a series of standards parallelly. So both retention time, which is seen in the x-axis, will be similar to the standards. So from that, you can easily identify the compound. So here, in this case, from the PA, 3-hydroxypropionic acid, there will be very high peak will be seen in the chromatogram of the PA case. But this one is for the MMA. Actually, it is for the mutase enzyme defect. Here, the methylmalonic peak is very high in this case. You can clearly see around the time is 9. So methylmalonic peak is very high in this chromatogram. Then next one is the whole exome sequencing means what? Actually, madam has mentioned finally that the mutation they have find out with the whole exosome sequencing. So The exosome sequencing means what? The first option is sequencing for the entire genome of the individual. And the second option is sequencing the gene of interest. And the third option is sequencing all the coding regions of the genome. And the option D is none of the above. So which one is the answer for the question number 8?
C. Sanchi, Almer, Adira, Prasna.
Yes, you are correct. So here exome sequencing means you are sequencing all the exon regions of the genome. That is the coding region of the genome. So by the next generation method of called next generation sequencing. And here you can see the result. Actually, madam have displayed in a particular format. Actually, genetics is a separate field. We have to learn a lot of from that, how to give the naming the variant, how to report particular variant. So here you can see that MMA is the gene, which is and the CBLA complement type has been reported as the mutant. Here you can see the MMA plus. Plus means this stands for plus strand or coding strand or non-template strand. You might be studied up when you are studying about DNA replication. What is sense and coding? Then you can see here in the bracket ENST. This is actually they have referred the gene sequences from the Ensemble. Ensemble, that is a genome browser. It is owned by the European Bioinformatics Society. Here ENS stands for the Ensemble. That is the number which they have given for the transcript. And the location of the mutation they have identified the exon 7. And the variant you can see here, C is for the coding region. And here 1069, 1077 deletion insertion and T. What does it mean? 1069 and 107 positions is deleted. Their two bases were deleted. Instead of that, one thymine is inserted there. That effect caused the change in the protein is the P stands for the protein. Here the alanine at the 357th position is changed to serine. And here one more nucleotide is deleted. That created a frame shift mutation. This FSC stands for the frame shift mutation. But here you can see TER20. TER20 means termination. There is a termination codon after 377th position. So it created a truncated protein. So this is the variant you're supposed to read up. And zygosity is homozygous and complementation is autosomal recessive, and it is classified as a pathogenic. Maybe earlier they have reported, and these classification sometimes pathogenic, likely pathogenic, maybe benign. That is totally based on certain recommendation from the ACMG, American College of Medical Genetics. So thank you for listening.
Wow, that was indeed wonderful. So we have learned a lot of diet, a lot about whole exome sequencing, and so many things. I think we should request Professor Girija Mohan ma'am to summarize organic aciduria for the students. Ma'am, please give us a word.
So good evening to one and all. From the gene transcript and exon to simplifying a general approach to inborn errors of metabolism, to simplify it, if you start from the history, because all the inborn errors of metabolism are autosomal recessive, so always inquire about consanguinity. And also from the antenatal history is also important, because even antenatal diagnosis is possible in many centers, and you can detect inborn errors of metabolism. Most of the time babies are born normally because placenta is able to excrete all the toxic products, except for few storage diseases. Then immediately after birth, depending on the severity of the deficiency, the symptoms manifest. If it is, for example, in this child manifested only by about 8 months. From the history, we can presume that this baby is not having a very severe defect, and thank God it is almost treatable with a dietetic restriction and all. But suppose it was very severe, in newborns, it is very difficult to distinguish from sepsis, meningitis, or if you cannot find a cause for sepsis or meningitis, or even HIE, hypoxic ischemic encephalopathy, unexplained drowsiness, maybe because of, so you cannot confirm the diagnosis of metabolic inborn errors of metabolism in all centers, but at least you should suspect. You must know all the atypical findings if you cannot find a cause. So sepsis, meningitis, drowsiness, HIE, all these can present as inborn errors of metabolism can present like this. And then another, as Dr. Vijaykumar already mentioned, persistent vomiting, which is you should never give antiemetics and keep an young infant with symptomatic treatment. There are so many causes. One is inborn errors of metabolism. Another is pyloric stenosis, which you should never miss. And also another important treatable condition is congenital adrenal hyperplasia, which comes in the differential diagnosis. All these differential diagnosis you should keep in mind. And then in the history, you must see if there is a history of unexplained sibling death. Yes, a sibling has died without any cause, and also there are any seizure disorder or mental retardation in the family, which for which a cause cannot be found out. So all these are important in the history. Then failure to thrive, in all cases, if you cannot find a cause, just like in this our patient, you must always, and the cause for this drowsiness, seizure, etc. is because of these toxic products are migrating to the blood-brain barrier is being crossed, and it will induce cerebral edema. That is one of the postulation for all these CNS symptoms. And also another differential diagnosis in young children, I told you, it is congenital adrenal hyperplasia. And then examination, it is not only the urine which is which has an order, it is even the sweat, which is just like in cystic fibrosis. If you the mother kisses a baby, there is salt there. Similarly, the baby has a smell, about which Dr. Cletus has already explained. So that is also very important. Hepatomegaly. In this patient, the patient had a liver dysfunction, and hepatomegaly and liver dysfunction is one of the clue to the diagnosis inborn errors of metabolism. Then another important, due to scarcity of time, limiting my discussion. Then regarding diagnosis, you must always remember, suppose the baby is normal. Suppose this baby is normal. If you investigate, baby will be normal. The investigations will be normal. Only during acute crisis, that is any stress which can induce catabolism, like fever, infection, or even immunization, can fever can cause this acute stress, and the symptoms can be if you can investigate at that time only, you will be able to diagnose. So you will be missing it if you are investigating whether the patient is in a normal state. That you should remember. And also, most of the investigations are done in blood and urine. When you collect urine, it is important that it must be transported as early as possible, or it must be refrigerated. And also some of the tests are done in very branded or any specific laboratories. Most of the laboratories will take up the sample, but you should be very careful. It must be sent to centers where it is being done correctly. That is also important. And also, you should never miss the patient, as this patient was diagnosed and the diet was restricted. That's the reason why baby is doing well now. So at the correct time, you must make the correct diagnosis, and do not make a delay in referring a child if you have a slightest suspicion of inborn errors of metabolism. I think that is in a nutshell regarding a general approach to a child with inborn errors of. And if you are advising diet, and it is very important you counsel the parents regarding this. It is you must sit, relax, and then counsel the parents regarding the condition, and highlight the importance of restricting diet and all, because in an 8-month-old baby, it is very difficult to restrict diet. It is practically not possible. We can talk all the theories, but it must be. 8 months. 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 8 mon just particularly pinpoint the diet which this 8-month-old baby can take. So that is regarding general approach to inborn error, baby with inborn errors of metabolism. Then I must thank Dr. Elizabeth for organizing this clinics. Then I must thank Dr. Vijay Kumar for actively involving. And then Dr. Sujin for an excellent presentation, Dr. Riju. And I think students are actually fortunate for this integrated classes, because when we were students, we were taught biochemistry. We never knew why we were learning all this. It is now only the integration has come up, taken up. So I think Dr. Riju has mentioned about the pathways and all, and Dr. Jyoti gave an excellent dietary advice regarding this patient. So thank you all, dear students, for participating. Hope you have learned something to carry home. Thank you very much.
Over to Vijay Kumar sir for his expert comments.
Okay, as a continuation of Madam was telling, the symptoms of an inborn error of metabolism can present sometimes abruptly, present, or it may be in the form of episodic presentations, or it may be in the form of a chronic illness, or sometimes it's a progressive illness. So, and also one point I want to stress is that it is mentioned by all of you. It is that a high index of suspicion is required for the diagnosis of an inborn error of metabolism. Most often, the inborn errors of metabolism, they have some usual symptoms. When you come across such a patient, you have to concentrate on the symptoms history, your clinical findings, and investigations for these, and you come to a final diagnosis. For this, you need not require all the knowledge that you have been taught in the biochemistry, because you may not be remembering all these cycles, Krebs cycle, citric acid cycle, all those things. But certain points may hint you this may be a case of inborn error of metabolism. Then sometimes these children may have a regression of milestones. Some of the milestones acquired by the child may get regressed. That also is another important point that you have to notice. And the onset of the disease can manifest usually when the food is introduced into the diet of the patient, and sometimes if it is a minor disease, it may go unnoticed. So it can manifest at various ages. It can manifest in the infancy, it can manifest in the older child group, it can manifest in the adolescence, and sometimes more rarely even in adulthood. In adult, sometimes these patients may manifesting with psychiatric symptoms. In psychiatric symptoms, then a stroke, then other neurological deficits, subtle neurological deficits, then all these manifestations. Sometimes some of these cases of, say, cerebral palsy that we were considering as a cerebral palsy, or some cases of, say, this motor neuron disease, or multiple sclerosis. All these cases may be inborn errors of metabolism, and such cases may be detected to be inborn errors of metabolism on further investigation. So we have got a very good presentation by Dr. Sujin, and a very good presentation by Dr. Riju, and especially the dietitian Jyoti, and I also again congratulate our madam, that is Elizabeth madam, for taking pains to conduct all these clinics. And one person behind this, all these settings is Dr. Jijo, our own Jijo. So he also to be congratulated, and thank you all.
Thank you so much, sir. Let us conclude and wind up this wonderful academic session. Thanks to all the speakers for enlightening us, especially Dr. Vijay Kumar KS, Dr. Jiju Joseph John, Associate Professor in Pediatrics, Dr. Girija Mohan ma'am, Dr. Sujin Mariam Thomas, Senior Resident in Pediatrics, Dr. Riju Matthew sir from Biochemistry, Dr. Cletus CC from Biochemistry, Jyoti ma'am from the Dietetics Department, for all of them for making this difficult topic of inborn errors of metabolism a difficult topic in very simple manner. All of them has described this very nicely. So all of the students, we ask you that the next every Thursday and Saturday we have such interdepartmental program. So we ask you that we should meet every Thursday and every Saturday. We also have a special thanks for to Dr. Maman Paul and all the webinar coordinators for setting up this telecommunication system. And so see you next week. Till then, goodbye.
Thank you. Thank you, Doctor.
Thank you, ma'am.